Hi, good afternoon or good morning, depending on where you are. Welcome to another meeting of the AEESP AJAR Future Faculty Seminar Series. And as you know, if you have been coming attending these meetings in the past, we want to celebrate the accomplishments of our students and postdocs, and also while also celebrating the diversity in our field. And we select 2 candidates every other week. We go through a small mock interview for faculty job positions with them. And then they give a 30 minutes presentation in this seminar series. Today we have 2 candidates, you have 2 outstanding researchers, and it's very interesting because these 2 they started with a non-engineering degree in chemistry and microbiology, but then they ended up doing their PhD in environmental engineering and applying their science to this field. And the first speaker that we have today is Dr. Ana Barrios. Ana Barrios did his bachelor's in biochemistry and master's in chemistry at the University of Texas at El Paso, and then went to do her PhD at the Arizona State University. And she actually is going to start a postdoc working with at the National Institute of Standard and Technology in June. So congratulations on your new position. She has been working on investigating the surface chemistry of and morphology of nanomaterials. And you can see she's going to talk about that effect on the development of sustainable engineering applications. And she has, last time I checked, she has 22 publications with several under review. And she has received multiple awards, including the Dean Award for Best Outstanding Dissertation. And she has done several outreach and volunteering activities throughout her time at the university. So she is going to present this work. And thank you so much for coming here. We are really looking forward to learn more about your research.
Well, thank you very much, first of all, for allowing me to be here today and give you a little bit of information about what I've been working on at Arizona State. So today I'll be talking about the structure-function relationships of nanomaterials and their applications for the development of sustainable engineering. And this is mostly focused on the work that I've been working on for my postdoctoral studies. So as one of my main research interests, I wanted to start working with water. And to do that, I looked into what are some of the nanomaterials that have been mostly applied for water treatment. And we saw that there are 2 main groups, namely the carbon nanomaterials and metal nanomaterials. Now, my work is going to mostly focus on the graphene-based nanomaterials, but I will also touch base on how silver particles can be applied for water treatment applications. Now, we know that graphene-based materials in general are widely applied in many different fields, but for our purpose in environmental engineering, we can use them as nanoencapsulants for plant protection, to increase crop yield as environmental sensors. But for the sake of my research purposes, I wanted to look at the properties of these carbon nanomaterials and use them for their antimicrobial properties and sorbent applications to remove pollutants from water. Now, when we first look at the wide array of graphene-based nanomaterials, we know that they have very different structural differences. Let's start, for example, with graphene, which is like the poster child of these graphene-based materials. And we know that it is a carbon-carbon sp2 bonded material with a sheet-like structure and a honeycomb lattice. But then we can functionalize that graphene material and give it different oxygen functional groups to its structure. And we can create the sister or the brother material called graphene oxide. We can also tune those oxygen functionalities and create a diverse suite of materials with different functional groups on their structure, ranging from like more oxidized to more reduced materials, as we can see in the picture. Now, because of these, we can also see that the graphene materials can have very different properties. And if we look at the literature, we see that a lot of these materials tend to vary in size, but also in oxygen to carbon ratio, which is an indicator of the extent of oxidation that these materials can have. And because of that, they can actually have very different functionalities and applications. And so the approach that we took for these work relies on understanding and looking at the nanomaterial structure and properties, number one, to see if we can apply these properties for a particular function, or and therefore establish these structure property function relationships, or if we can look at these properties and see what are their hazards, if they can be some sort of structure property hazard relationships. Now, so for the sake of this presentation, I'll be giving you examples of how we have elucidated, how we have evaluated these relationships, but also I'll be talking about a third example where we try to combine both of these relationships and try to find a way where we use the hazard property of a material while also enhancing the functionality of these properties. So the first project that I'll be talking about is the structure property toxicity relationships of graphene oxide. And we're going to be looking at how the surface chemistry of different graphene-based nanomaterials impact the mechanisms of interaction with bacteria. Now, to understand these a little bit better, we need to know how these materials are being used as antimicrobial agents. And so if we look in the literature, we see that there are two main types of mechanisms of interaction. We know we can have a physical mechanism where the graphene oxide tends to adhere to that bacterial membrane and kind of like adhere to the lipid bilayer. But because we also know that the sheet has very, very sharp edges, the materials can also cause these membrane puncturing. It's also called as a nanoknife effect. On the other side, we can also have these chemical mechanisms where we can create oxidative stress, generate oxidative stress. And that's usually evaluated and observed as an increase in reactive oxygen species production. So for this work, we are going to be working with a commercially available graphene oxide material, which we're going to call as receive GO. It's a very oxidized, highly oxidized material that we will then thermally anneal at different temperatures, namely 200 all the way to 800 degrees Celsius. And this thermal annealing basically reduces the material in a systematic way. And upon surface characterization, we see that the oxygen content decreases as we increase the annealing temperature. So now we have 4 different materials that are systematically reduced. And we're going to continue to analyze these with how they interact with bacteria. Now, the first thing that we wanted to do was to generate these dose response curves. And we did this by integrating bacteria, in this case, E. coli, with a wide array of concentrations of these materials. And we see that each of these materials generate a different response at different concentrations. So what we wanted to do to kind of like narrow down how these interactions happen, we calculated the effective concentration at which 50% of our bacteria generated a response towards the material. And in this case, we see a very strong linear relationship between the EC50 value and the extent of oxidation of our materials, where the more oxidized material, in this case, ARGO, usually requires a lot more material for it to reduce bacteria viability by 50%. And the most reduced material requires less. Now we're going to shift the question that we have. We know that these materials are showing some toxicity, but in order for us to change the question from how, from are these materials toxic to how these materials elucidate that toxicity, we wanted to do a set of fluorescent dyes-based assays. And we did this by using 3 different fluorescent dyes, each of them kind of like indicating different biochemical responses on the bacterial cells. And for example, we have fluorescent diacetate, which is usually an indicator for membrane integrity and esterase activity. We have BODIPI, which is an indicator for lipid peroxidation. And we have dihydro dichlorofluorescent diacetate, which is an indicator for reactive oxygen species generation. Now, upon the first results that we see is that the as received GO, which is the most oxidized material, has the highest reactive oxygen species generation, and this actually leads to oxidative stress. On the other part, we see that the thermally annealed materials actually show an increase in FDA signal, and that is attributed to perhaps more interactions between the cell membrane and the materials, therefore leading to kind of like a wrapping mechanism from the material to the bacterial cell wall. Now, from this work, we can see that the graphene oxide surface chemistry has a really big impact on the antibacterial potency of these materials. We see that especially in a more clear way when we calculate the EC50 values, and we see that the antibacterial activity increases as the materials become more reduced. And we also see a shift in mechanism where we see that the more oxidized material displays a chemically driven mechanism, as we displayed by an increase in reactive oxygen species generation. But we also see that the thermally annealed materials display a physically driven mechanism, as we saw with the fluorescent diacetate dye, where we show that there's perhaps a little bit more interactions with the membrane overall. So now we have already shown you how we can look at the nanomaterial properties to establish this structure property toxicity relationship. Therefore, I'm going to continue by showing another example where we look at the structure property function relationships. Now, this next work deals with how can we remove bromide using a graphene silver composite. We're going to be looking at the role of particle size and carbon structure. So the motivation behind this work is we know that in water, we have bromide, which is ubiquitous. It's a ubiquitous element. However, when we have natural organic matter and a type of disinfectant, namely chloramine, hypochlorite, any type of disinfectant, we tend to form these brominated disinfection byproducts. And throughout the water treatment process, we don't necessarily tackle a particular technique to remove these disinfection byproducts, and they're actually very, very genotoxic and cytotoxic to multiple species, including humans. So what we wanted to do is we wanted to tackle the removal of the inorganic component of these brominated disinfection byproducts. And the way that we wanted to do that was, well, we want to look at how can we remove the bromide by precipitating it out of the water. So we looked at the different bromide species that could be precipitated, but we saw that we didn't really want to use mercury or copper or lead, because that really, we know that these elements are very toxic. And so we wanted to look at something that has a high affinity to bromide, but it's not as toxic. So we actually decided to use silver. We know that silver has a very high affinity to bromide, and it tends to form this silver bromide precipitates upon contact. One of our previous work actually dealt with comparing this activated carbon with compared to graphene oxide. And when we functionalized these with silver ions, we saw that as we increased the complexity of the water, the removal capacity of the nanocomposites actually decreased. But if we were to compare these two materials, we see that the graphene oxide has a higher removal capacity than activated carbon. And these kind of like led to the motivation of our project. We know that graphene oxide functionalized with silver salt is a good material to remove bromide from water. However, what if we were to use silver nanoparticles instead of silver salts. So the whole motivation is, let's compare a salt versus a nanoparticle-based composite. We have different experimental conditions, and we started by working with 3 different types of graphene oxides. These are all very structurally different. The names come from the synthesis method that were used, and basically the main difference is that they have different extents of oxidation. Stoudemire is a very reduced graphene oxide, whereas tung is a very oxidized graphene oxide. And so we wanted to synthesize the silver nanoparticles in situ. So we used a salt precursor, namely silver nitrate, and we reduced these particles by using different concentrations of sodium borohydride. Now, so we have composites with graphene oxide and silver salt, graphene oxide and silver nanoparticles of different sizes, but we also wanted to see if we actually needed the graphene or the carbon support on this composite. So we tested commercial nanoparticles of different sizes to see if there's actually a size-dependent effect for bromide removal. So when we look at the first set of results, looking at just model water, no competing ions present, we see that silver salt has a very high efficiency at removing bromide from water. When we start looking at our different composites, let's look at our silver salt composite. We see that that efficiency reduces, but when we look at the nanocomposite and the silver nanoparticles, we see that that efficiency actually is increasing. And if you look at the silver nanoparticles, we actually see kind of like a size-dependent effect, where the smallest particle has the highest bromide removal capacity, and the largest particle has the least. Now, we wanted to go a little bit further, and we wanted to see if the competing ions, as we increase the water chemistry complexity, have an impact in this bromide capacity removal. So, and we do see that, in fact, if you look at the silver salt, that great removal capacity that we had in model waters is completely hindered by the presence of competing ions. And we did this in a synthetic fresh water system, where we know that we have chloride sulfates, which will also interact very strongly with silver, and therefore reduce the ability to form these silver bromide complexes. When we look at our nanocomposite, we see that that removal efficiency is actually very high, and it's significantly higher, especially compared to my silver salt alone. And when we look, And when we look at our silver nanoparticles, we see that that removal efficiency is actually also keeping that high effect, right? And we also see that there is a size effect with silver nanoparticles as well. So let's talk a little bit about what's happening. And we have theorized 2 main mechanisms to remove bromide using the silver composites. The first one is, let's suppose we have bromide present in water. Upon the addition of silver nanoparticles, we know that the silver nanoparticles can dissolve into, or start dissolving into, silver ions, which will then form the silver bromide precipitates. However, we have another mechanism that can come into play. Let's again suppose we have this bromide ion in water that will then come in contact upon the addition of silver nanoparticles. But then we suspect a direct interaction between these bromide ions and the silver nanoparticle surface. And so in order for us to test these, and again forming the silver bromide precipitates, but in order for us to test these, we needed to do this set of anoxic experiments just to remove that dissolution effect and make sure that the effects of bromide removal were actually coming in because of that complexing between the silver and the bromide. And so we saw that there is, in fact, if you look at the dashed graphs, we see that the removal efficiency of the materials actually increases in anoxic experiments. And that's because we attribute that to the direct interaction between the silver nanoparticle surface and the bromide. And that we attributed to the halide etching, which is actually observed when we're trying to synthesize silver nanoparticles. We use halides, namely chloride, iodide, and bromide, to tune the shape of our silver nanoparticles. but this is the first time that we have actually observed this application for the halides in water treatment. So another thing that we found is that bromide etching is not necessarily oxygen dependent, and that's why most of our materials have a similar bromide removal capacity. When we compare that to chloride, where we actually see less removal capacity with chloride ions present. The other thing that we wanted to test was, and as I mentioned, something that we really want to make sure is that we are not removing one contaminant by adding another one. So the EPA has the secondary maximum contaminant level for silver, and it's set to 100 micrograms per ml. However, when we do this jar test, which simulates whatever happens in water treatment in terms of flocculation, coagulation, and sedimentation processes, we see that these nanomaterials still have some silver remaining in solution, but it is definitely below that level set by the EPA. So that's actually really good. If we take a closer look, we see that the nanocomposite, the graphene oxide silver nanocomposite, actually is the most effective at slowing down or preventing that silver to be released into the water stream. And that's actually contributed to the fact that it has this carbon support, which supports the fact that this carbon base, because of its micron size, can be easily coagulated and settled down after the addition of alum. So based on this work, we can conclude that the graphene oxide functionalized with silver nanoparticles is efficient at removing bromide from complex water matrices. However, we also see that competing ions, namely chloride and sulfates and phosphates, will definitely have an impact at how these materials remove the bromide from the water. But some of the mechanisms that we see is that we can actually have direct interactions between the bromide and the silver nanoparticle surface, creating this etching ability. We also saw that the carbon actually facilitates the removal of silver. And so therefore, now we have, I have talked to you about these different relationships of the materials. And so what I want to show you next is how can I combine both of these relationships and make a material that is inherently hazardous? How can we apply this hazard property and make a functional and provide a functional outcome out of it? And so for that, I'm showing this third project, which is where we're trying to prolong the antibacterial activity of nanosilver-coated membranes through partial sulfidation. Now, this project was motivated because we know that in water treatment, silver nanoparticles are widely used for their antimicrobial or antibacterial properties. And we tend to functionalize these membranes with silver nanoparticles to reduce biofilm formation. However, one of the main drawbacks of these is that we tend to have dissolution of the silver nanoparticles over time, and therefore we might have to recharge the material as we pass the water, as we operate the system. And so we chose to use sulfidation. And why sulfidation? If we look again at the type of silver species that we can create, we saw that if we have sulfur present in water, we create these really, really strong sulfur sulfide composites. And we know that the sulfur will actually have a really high affinity to silver. So we thought this would be a good approach to try to slow down the release of silver by creating these little silver sulfide layer. So what we did first is, we wanted to create this in situ synthesis of silver nanoparticles on a reverse osmosis membrane. We also know that if we were to leave the nanoparticles alone, we expect a rapid dissolution of silver nanoparticles into silver ions. So we added this sulfidizing agent, which is called, which we use sodium disulfide to create these partial sulfidation, this partial layer of silver sulfide on top of the nanoparticle surface. And that will actually lead to a slower release of silver ions. And that's actually a really easy functionalization, we see that, for example, our control membranes are nice and white. Upon the addition or the synthesis of silver nanoparticles, they turn brown, and upon the addition of our sulfidizing agent, they turn a different color as well. So we can do this even just by visual inspection. The first thing that we wanted to make sure is that the sulfidation really slows down the silver release. And so we tested the silver nanoparticle, and we saw that the silver release was about 138 nanograms of silver per centimeter square per hour. But upon sulfidation, we see that that silver release dramatically decreases up to 20 nanograms of silver per centimeter square per hour. And that actually tells us that indeed the sulfidation step helps us slow down that silver release as we operate that membrane. So the next thing that we wanted to see is, again, because we know that silver is an antibacterial, we wanted to see if we can actually apply this in a reverse osmosis bench scale system to see if we can reduce biofilm formation. And so we operated this system for about 24 hours. We loaded the membranes into each of these chambers, and we saw that the unfunctionalized membrane was the first one to decrease in flux. We attribute that to the formation of biofilms on the membrane surface. But if we test the sulfidized particles, we see that the flux, it's still pretty good, and it'll take longer for the membrane flux to decrease. Not only that, but we also saw that when we took the membrane out of the module and we test how much silver was remaining on that membrane surface, we saw that for the silver nanoparticle, a lot of the silver was actually depleted. But upon sulfidation, we see that the thicker the layer of this silver sulfide, we see that there is, in fact, more silver still remaining on the membrane surface, which can actually help us improve the lifetime of these reverse osmosis membrane. So from this project, we have concluded that sulfidation can actually decrease the silver release by over 80%. And we know that the slower silver release rate is more important for dynamic biofouling resistant. And this is because we want a constant, but also slow release of silver ions to remove biofilms more efficiently in a reverse osmosis module. And also something that I wanted to highlight is that in fact, sulfidation is a simple and yet effective way to prolong the lifetime of anti-biofouling coatings. So for the concluding remarks, I have shown you 3 different projects. The first one where we try to establish these surface property hazard relationships. And we have seen that as we decrease or as we change the surface chemistry of our materials, we actually have different interactions with bacteria. The other project basically tells us that if we were to create a nano composite, right, and a graphene oxide composite, and we were to functionalize that with silver nanoparticles, we can increase the performance of these materials at removing a pollutant from water. And last but not least, we have shown a merged interaction between these relationships, where we see that, for example, if we were to sulfidize silver nanoparticles on a reverse osmosis membrane, we can slow down the silver release while at the same time maintaining that antibacterial property that we want for biofouling control. Thank you so much for listening, and I'm open for any questions.
Great, thank you so much. It was an excellent presentation. I see that we already have 2 questions in the chat, and they have to be pretty quick because we are in the 12:30. So I was looking at, and I think that the first one on raising the hand was Bhavani Cherukupali, and then Mohamed Atiyah. So Bhavani, if you want to go first. And you should introduce yourself first and then ask your question, please.
Okay. So hi everyone, my name is Pavani. I'm a postdoc at Imperial College London. And thank you so much for the informative presentation, Anna. And I have a question. I have a question around slide 12.
Okay.
I'm trying to understand like how you use this fluorescence technique to identify the biocidal mechanism between oxidative stress and the annealing process.
Okay, how did the fluorescent dyes work, correct?
Yes.
Okay,
Yes.
so I actually have a little slide here that just to explain these really quick. So basically, we start with the H2DC-FDA, which is the reactive oxygen species indicator. And this is a permeable dye that crosses the bacterial cell wall. But upon esterase enzymes present on the cell wall, on the bacteria, we actually create these fluorescent compound, the DCF compound, and that's representative of the different reactive oxygen species that are present on the cell. So that's how we can see the signal on the reactive oxygen species generation 1. Now for the FDA is an indicator for esterase activity. So if we have more esterase activity, then we can generate a fluorescent response for that particular dye as well. Now these two dyes, H2DC-FDA and FDIA, are dependent on each other. So there's a relationship between those two. Does that help illustrate your question?
Yes. And how could you know, like it is you? I think after the slide 12, you mentioned two mechanisms. One is oxidative stress. This slide answers that, and the other one is the annealing process. Annealing is the mechanism of bacterial death. Something.
So the annealing is related to how we created the material. The thermal annealing just basically helps us create materials with different surface chemistries. But really, when we look at the mechanism, we're dealing more with two types of mechanism. The first one that we saw was for the oxidized material, where we see a higher reactive oxygen species production. And so therefore we say that ARGO tends to have this type of mechanism interacting or happening within these bacteria material interactions. We also did further testing. We're not just basing our conclusions on just these fluorescent dyes. I know from our collaborators from the University of Pittsburgh, they also helped us with glutathione assays. And we also did some microscopy studies to confirm that these interactions between physical and chemical mechanisms observed are actually supported.
Good, thank you. And by the way, which cells did you tested for these studies?
Sorry?
Which bacteria did you test?
This is with E. coli.
Thank you. Oh, sorry. I have one more follow-up question.
No, I think that I think that we are running out of time, but if you stay until the end, we'll have that 15 minutes networking after Sarah's presentation, so you can stay. And we have Mohammed that was going to ask a question. If it is quick, yes, please go ahead. If not, if we can do all these questions at the end.
Yeah, I actually have a meeting, so I will just ask my question very quick.
Okay.
Thank you very much for the presentation. I'm Mohammed Atiyah. I'm a research associate at Northwestern. Both of your work is very interesting because I did both research before. The first thing about the disinfection using nanomaterial, this is something that I find interesting, and I don't know whether you considered it before or no, is the regrowth of bacteria after such disinfection method. Have you observed or monitored the regrowth of bacteria and how significant it is compared to chemical disinfection methods? This is my first question. Second question is about the bromide removal project. I also did that, and I had my composite like with silver chloride. So the question is about how is the challenging condition that you tested? Like, what was the ratio of chloride to bromide? Did you test like sulfate? Because I mean, I did that, so I know how challenging it is if you have just silver, because if you have any other anions, it will just precipitate as other anions. These are my two questions. Thank you.
Definitely. I feel like for the sake of time, I have a faster response for your second question, and I'll just show you this table. The conditions that I used for my more complex water matrix was using these freshwater recipes. It's a synthetic freshwater that we get from our engineering research center. And we see that we did test all of these other competing ions that have really strong affinities with silver, namely, for example, sulfate, phosphate, and chloride. If we look at the concentrations of chloride present, it's about 75 milligrams per liter. And the concentration of bromide that I used was 200 micrograms per liter. So the concentration is orders of magnitude higher, which is basically what we see in a realistic water matrix. We have much more chloride present than bromide. And that definitely impacts how these materials remove bromide from water, because they do seem to have a decrease in the removal capacity.
Okay, yeah, we can chat later. Thank you.
Great, thank you so much, Dr. Barrios. What an incredible talk, and we appreciate the Q&A as well.
Thank you very much.
Thank you. So it is my honor to introduce our second speaker today. So Dr. Sarah Ben Mamar. Dr. Ben Maamar comes to us with a background in microbiology and microbial ecology, with postdoc experience in environmental engineering. She completed her PhD in the University of Rennes in France, with research related to the characterization of groundwater microbial diversity and functions in a gradient of agricultural nitrate pollution through metagenomics and geochemistry. She is currently at Northwestern University and has done postdocs with Dr. Louis Nunez Amaral and Dr. Erica Hartman. So bridging the gap between microbiology and engineering, with really interesting applications for data science, big data, using high throughput sequencing. And so I'm very excited to introduce Dr. Ben Maamar without taking any more of her time. The floor is yours.
Thank you so much, Dr. Johnson and everyone for the nice introduction and for being here. It's so exciting to see so many faces. So today I'm gonna try to give you an overview of the span of my research, from healthier buildings to reproducibility of bioinformatic pipelines. And I'm gonna try to touch upon the challenges and solutions that I faced and found. So just as a starting point, this is my presentation outline. I will start with like a few concepts about microbial ecology, and I follow up with three main research topics that I worked on. So let's start with some few concepts. So why do we care about microbial ecology? Well, first of all, what is microbial ecology? This is sometimes named environmental microbiology, and this is defined as the ecology of microorganisms, including both their relationship with one another and with their environment. It includes mainly eukaryote, archaea, bacteria, and viruses. And in addition, we like to study them because they have a very important role in biogeochemical cycles. They are omnipresent, even in very extreme environments, and they constitute just by themselves significant carbon sink, as they represent about 10 to the power 30 cells. How do we study that? Well, it started with a lot of with cultivation. You have an example here of a petri dish, for example, to cultivate bacteria. And now more and more, we're using more molecular approaches. An example is the 16S rRNA sequencing, which allows you basically to decipher which bacteria are present in a specific environmental sample, for example. You can also use metagenomics, which is, or whole genome sequencing, if you are interested in knowing what those bacteria can eat or respire. You can also use metatranscriptomics, single cell sequencing, metaproteomics, metabolomics, in case you really want to know what this microbe is eating at this time, for example. And you have obviously microscopy to visualize them. You have also flow cytometry to count them. Okay, so microbes are very fun to study because they both have deleterious and beneficial effects. Example of deleterious is, for example, biocorrosion. As you can see here, they're also responsible for food spoilage, antibiotic resistance, or any drug resistance, actually. COVID-19 is an excellent, unfortunate example of deleterious effect. And they can, bacteria, viruses, they can mediate a lot of different infections in humans. They also have obviously beneficial effects. One of them is cheese, for example, probiotics, also gut microbiota. If you have good gut microbiota, you will help your brain to function better. They also are very helpful in purifying water. And they're also obviously playing a huge role in plant growth, but they can also cause diseases in plants. So I hope I convinced you like that this is pretty fun to work on. Currently, we have still some challenges in microbial ecology. Some of them are fundamental. Some examples are, for example, understand and predict interactions between microbes. Also predict microbe functions, or understand how microbes resist to specific pollutions or perturbation in the environment, and etc. You obviously have also applied challenges. One of them is antibiotic resistance, for example, or environmental bioremediation. We can use bacteria to help in the remediate polluted sites. And they are obviously also very useful for human health, as they are very beneficial to us, such as probiotic, for example. And they play a huge role in mitigating global warming, etc. So I hope I convinced you that microbial ecology is worth looking at and studying. And let me now start a bit more with my research and give you a little bit of background about what I did on groundwater microbial community and nitrate pollution. So this is basically my PhD work that I did in France. My PhD work happened in the Brittany region that is located in France. And this is on northwest of France on the Atlantic Ocean. And this region is actually well known for its intensive agricultural activities. Actually, Brittany region is the second producer of pork meat in Europe. So pretty heavy activities over there. It is also a region that has a lot of water, and they have specific geological structure that allows water storage that are called aquifer. And the problem is that the nature of their aquifer there in Brittany makes it very vulnerable to the infiltration of pollutants such as nitrate, for example. There is a lot used obviously in agricultural activities. So I also mentioned to you that microbes are very crucial in water purification. So we can study microbes in those specific environments to understand how we can use them to potentially remediate this nitrate pollution that we have there. So the two main questions that I investigated during my PhD were first, how does nitrate pollution impact the natural microbial communities? And how does, what does limit the denitrification in situ, basically? In order to do that, I focused on 3 distant aquifers that you can see here in these 3 cities. And for all of those aquifers, I sampled both superficial water and deeper water. This is a cross-section of an aquifer. Okay, so once I have sampled those groundwater, first of all, I obviously characterized their chemistry because I want to know which nutrients are present over there for microbes to survive. And I used like different techniques, ICP-MS, IC, ionic chromatography, total organic carbon analyzer. And I also used analyzed the dissolved gases into those groundwater because those gases allowed me to decipher how long this specific water that I sampled stayed into the aquifer. This is what we call the groundwater residence time. In addition, I obviously characterized the microbial communities, and the techniques that I used were mainly molecular biology, DNA, RNA extraction, RT-PCR, and etc. Obviously sequencing, library preparation. And obviously when you produce sequencing data, well, you need to analyze them and do some bioinformatic analysis. So I mainly focused on 16S RNA, metagenomics, and metatranscriptomics. And in a nutshell, in terms of results, what we got through this study was that basically the biodiversity that we see in our different groundwater samples is a reflection of the groundwater age. So here you have like this figure, for example, showing you different networks. So each node here shows you a microbial taxa, and each edge in between those nodes shows you a correlation. That means that those microbes tend to appear in the same site at the same moment. the shapes indicates you that those microbes were found in specific area. So it indicates the sample they come from, basically. And you can immediately see that whatever site we looked at, the old groundwater in terms of biodiversity, microbial diversity, they always look alike. And they were really dominated by specific bacteria. On the other hand, recent groundwaters, when you compare them with each other, they also really look alike. And it didn't matter where the actual site they came from. So in a nutshell, what we found is that in old groundwaters samples, we had a high predominance of iron oxidizer, Gallionella families, an excellent example of them, but also a lot of sulfur oxidizer like Sulfuricella here. In the recent groundwater, we find many more nitrate-reducing bacteria, such as Firmicute oxalobacteriaceae. And so in conclusion, we basically figured that groundwater residence time is an important driver for this biodiversity, and it can be used to assess vulnerability to pollution in each groundwater bodies. We also realized that the nitrification, especially in the recent groundwater that were more exposed to nitrate pollution, is unfortunately limited by the organic carbon availability. And one route, for example, to try to tackle this challenge is to try to supplement those groundwater with organic carbon and see if we can foster better this denitrification and removal of nitrate. Okay, this was for my PhD work. Let me now switch to what I did during my postdoc at Northwestern in Civil Environmental Engineering, where I had the chance to focus on the indoor microbiome and the associated risks. Few facts about indoor microbiome to start with. Many of us live in urban areas, and in urban areas, we spend about 90% of our lifetime indoor. And in 2050, it is projected that 66% of the world population will live in cities. In addition, there is this recent study that basically found out that 90% of US homes presented at least 3 allergens, and some of them were really present in high concentration in our homes. And you probably obviously also heard about this huge challenge that we're facing right now, which is the global warming. And we expect with this global warming to have way more particles in suspension in air in the coming years. In addition, within our homes, dust tend to concentrate a lot of particles, chemicals, but also bacteria. And we know by now that bacteria are also very challenging because of the problem of antibiotic resistance, which are responsible of a lot of death, both in US and in Europe. So the research question that I was tackling during this postdoc experience was basically, what is the potential for antibiotic resistance genes dissemination within dust microbial communities in presence or absence of antimicrobials, and trying to understand what is the risk associated to this antibiotic resistance gene dissemination and how to make healthier buildings. And in a nutshell, again, the way we proceeded was first to sample dust from 63 different buildings, from which we produced 167 dust bacterial metagenomes. From there, we also, from the same samples, assessed the concentration of antimicrobials reported in each of our sample. So we looked at triclosan, trichlocarbon, and a bunch of parabens. And obviously, with all this metagenomic data, one of my tasks was to quality control them, assemble them, and annotate them in order to screen for antibiotic resistance genes, mobile genetic elements, and also mobile antibiotic resistance genes. And about the methods that I used to address this they were mainly like relying on metagenome assembly, genome assembly, long read sequencing, for example, through MinION nanopore technology, and also conjugation and minimum inhibitory concentration assays. And so just to give you like some of emblematic results that we got through this study, the first thing that we found out was that in terms of mobile genetic element present in dust metagenomes, we mainly found that most of them were transposon and then followed by integron and then plasmids. Then when we looked at the mobile antibiotic resistance genes that were associated to those mobile genetic elements, you can see here on the y-axis different mobile antibiotic resistance genes, and on the x-axis different mobile genetic element type. What we found was that Most of our mobile and antibiotic resistance genes were associated to transposon. And interestingly, we also found that some of those antibiotic resistance genes, for example, this GADB gene here encoding for resistance to aminoglycosides, was specifically found on plasmids, while some other antibiotic resistance genes were mostly found, like for example, coding for beta-lactamase resistance or tetracycline resistance were mainly found on transposon. The thing is that when we did more work in our lab through cultivation, we realized this thing. A lot of mobile antibiotic resistance genes are present in our dust metagenome, but they are not first always expressed, and they are not also always transferred from one bacteria to the other. So the main conclusion of our work was basically that we really need to take into account these effects into our exposure assessment and risk assessment. And so just to finish about this topic, when you look at your home, imagine when you go, for example, wash your hands, you create a bunch of actually your aerosols that contains a lot of those chemicals. And those chemicals very often end up in your dust. The dust, as I mentioned, also contains a lot of bacteria. And so we suspect that some of those chemicals might trigger some antibiotic resistance genes transfer, but we have a lot of other factors that comes into play. For example, if you're on a pet, or if you open your windows more or less frequently, all of this can impact this indoor microbiome, and a lot of research is still needed to be done on this topic. And finally, I'd like to also introduce you to my most recent work that I'm actually currently doing here at Northwestern in Chemical and Biological Engineering, which is about the reproducibility of bioinformatic pipelines. So let me introduce you to this topic with this very interesting paper that was published in Nature in 2016 by Baker. And in this study, the author basically interviewed like about 1,500 researchers from different fields and asked them this very simple question. Do you think there is a reproducibility crisis in your field? And you can see their answer. About 90% of them said yes, there is a significant crisis. Then they asked them, the same researchers, have you personally failed to reproduce an experiment, either your own or either someone else? And you can see the results here across all the disciplines. You can see that, first of all, this problem spans a very high amount of disciplines, but it is particularly true for chemistry and biology. And actually, in biology, we use more and more of those next generation sequencing technology because their costs have really drastically decreased. You can see that since 2008, the cost has been stable. And at the same time, obviously, you see a huge increase in the utilization of this next generation sequencing technology in biological studies, and now most of our data in our databases are coming from those technologies. I cannot start talking about biology without making sure that we all agree on this central dogma in biology. In biology, we assume that from a DNA molecule, you get a transcription that ends up producing RNA molecules that then get translated into an amino acid sequences that ultimately forms our protein. The thing is that from in your cells, you all have the same DNA molecules. Like all your cells have exactly the same DNA molecules, but despite that, you can still, your body is still able to produce very different type of cells. For example, blue cells here and an orange cell from exactly the same molecule. How is that possible? This is possible through this particular step that is called transcriptional regulation, through which basically your cellular machinery basically will emphasize the expression of one gene versus another depending on your cell type. For example, this gene 1 is expressed here in this blue cell, but it's not expressed in this orange cell. And what we call like the comparison of this expression profile between cell type, for example, is called transcriptome profile. These transcriptome profiles, they are obtained through RNA sequencing technology. But what is that? In a nutshell, when you do RNA sequencing, you start basically by extracting all the messenger RNA molecules from a cell. Example here, you have different RNA molecules coming from a red gene, an orange gene, and a yellow gene. For technical purposes, you have to fragment those molecules and then basically put them into the sequencer and almost magically. It's obviously very simplistic. You get your raw data, which look like that, basically a bunch of ATGC sequences. But the thing is that when you look at this data, you cannot say much from a biological point of view. So what you need to do is to go through a computational pipeline to make your data more understandable and interpretable. This computational pipeline starts with this first step that is named quality control. And during this quality control step, you have to filter out basically the sequences that are low quality, also remove part of those sequences often at the end that doesn't meet the quality threshold. That is a consensus now in the field. Once you have done that, you can use your clean sequences to map them back to a genome of reference. Here you have a piece of genome of reference that contains a red gene, an orange gene, and a yellow gene. And you basically use this reads, that we call them reads, to map them back to each of the different genes that you have in your genome. Once you have done that, you can then do a counting step. So basically the counting step is just referencing, for example, here in this red gene, you have like about 5 reads that were mapped to this gene. So while on the orange gene, you have only 4 reads. So that means that this gene red here is more expressed than this gene 2 here, gene orange. So it's really like seemingly simple. The problem is that right now we have multiple algorithms that are available for each step. So you start with your raw data. Some people, some scientists will do the quality control, some others won't. You have plethora of algorithms for both alignment step, counting step, and the differential gene expression analysis step. If I just look at the different combination with these softwares that I'm presenting you here, you have already 84 variations. The problem is that each of those software, they have different modes, they have different parameters. And sometimes people use 3 different versions of their genome. At the end of the day, you end up with about 54,000 variation of pipelines. So if nobody, or if the person who processed the data doesn't document correctly how the data was processed, it's almost impossible for somebody outside of the study to retrieve how your raw data was processed. So what we did in this work was basically to look at different parameters. For example, if I take the HT-seq software here, it has different parameters. So what we started to do first was to look at, if I tweaker a little bit this parameter, I use this number versus this number, how much difference do I get starting from the same original data set? How much difference do I get in my result? This is the first thing we did. And the second thing was to look at, okay, if I like try different combination of those softwares, I can create different pipeline variation. Same question, if you start from the same original data, how similar or different are your results using one pipeline versus the other? And this is what I'm gonna show you. Here is just obviously just a sample of what we did. So you have like here this correlation matrix that shows you, for example, different counting software here on the x-axis and the y-axis. And the colors indicate how much in agreement are the results of one software versus another. So if it's red, that means it's really in agreement. If it's blue, that means the results are in disagreement. You can see that, for example, if I look at HT-seq, it has 2 modes, a mode A and a mode B and C. So if I compare the 2 different modes of HT-seq, you can get like pretty good correlation. Results are pretty good in pretty good agreement. But if you look at HT-seq and cufflinks, for example, it's still in agreement, but the correlation looks much more blurred, as you can see here. Now, the thing is that if you compare HT-seq to feature count, you have opposite results. So they don't agree at all. This is a first, one of the first problem we identified for reproducibility. The second thing, as I mentioned, is that we also compared the different pipeline versions that we, using a same original data set, and we looked at how different were our results with the different pipelines. And what we found is that in most of the pipelines, so 65% of the genes were found not differentially expressed whatever pipeline we used. So we are happy with that. It looks pretty good. This is pretty standard. 5% of our gene looked like they were all positively expressed whatever pipeline you used. Same here, we have 11% of our gene were found like always negatively expressed whatever pipeline variation we used. The problem is that we found about 19% of the genes that were sometimes predicted as positively expressed in one pipeline and negatively expressed in the other pipeline. So the thing is that what I'm gonna conclude here is that we have a huge lack of methods, results, and inferential reproducibility for computational pipelines used in RNA-seq, and that's why we really need computational standards on how RNA-seq data should be processed and analyzed. And with that, I'm gonna thank everybody, and particularly my collaborators. I got very lucky to work with all of them, and I'm happy to take any questions. Thank you very much.
Thank you so much, Dr. Ben Maamar. Do we have any questions from the audience? I see, oh, it's Lucia in the chat.
I have
Anish.
a question.
Yes,
yeah,
please.
So I think you'll have a hard time convincing everybody to do a good job, right? So we can maybe also think about to follow a recommended pipeline. My question to you is, as a user, when I read these studies, when you read these studies, how do you judge that, well, this person used this pipeline and I'm okay with their paper because they did this, this, and that? So how do you convince yourself that this study is acceptable to you and you feel like it's a good study?
So that's a great question. Thanks for asking this. So we're trying not to be judgmental about the pipelines that has been used by different authors. Really the main aim of this work is to really try to educate people on the fact that the choice they make on how to analyze their data is really important, and it's introducing bias in their results. And we really want to make people be aware of those biases. So obviously, the best pipeline to use, it really depends on the quality of your data and a lot of also other parameters, like the nature of your data set as well. So what we are trying to provide here is basically a tool that through which people would be able to enter their input data, their data set, and get it analyzed in different ways. And having this output, the scientists will be able to judge how much difference they can get between different pipelines. And so how much can they be confident in the trend they see in their data set. So it's really trying to help people in having a more confident interpretation of their results, and really have an eye on what is really okay, a strong, solid signal versus what is maybe spurious or an artifact, basically.
Sounds good. I'm sorry, I forgot to introduce myself. Manish Kumar, University of Texas at Austin.
Dr. Ben Maamar, in thinking through how we can move to a system that better considers this reproducibility, for the funding agencies, should they approach this from a kind of a top-down perspective, where we need to develop the tools that everyone will use and the oversight necessary to kind of call out better or worse behaviors? Or is it more of a bottom-up where every single project needs to consider this and justify their choice of pipeline before publishing results?
Thank you for the question. I think it's probably both sides. So I would say that the first thing is that the ANIH, for example, is really involved into this initiative of trying to provide better standards. Well, it's mainly the biological field, particularly the medical field, actually. They use heavily RNA sequencing technology. And so they're really pushing to try to have some benchmarking work that is being done on different tools. Obviously, tools evolve, new ones appear, and old ones disappear or tend not to be used anymore, hopefully. So really the idea is that there is a push in terms of assessing, doing some benchmarking work and assessing biases in the choices we make as scientists. So I think that obviously, I think the main goal, I think, from this initiative is really to make people aware, realize that they have to be critical when they look at the results and not take it for granted, which is very easy to do, to be honest. And I'm including myself in the group. It's because you create so much data, you generate so much data, and you are it's very attractive, you know, and you are like very excited, you want to interpret all of it, but sometimes it's just not the best thing to do. And sometimes you should maybe step back and say, okay, what can I really be confident about here? What can I really use, and what is more like maybe or maybe not? And that would help in also hopefully the scientists in deciding, okay, this hypothesis is interesting from this data set, but maybe we need to do a better job at certifying or like attesting basically.
Yeah, that's broadly applicable advice for all domains. I think we all need to take that to heart. Alexander, I see that your hand is up. It's maybe one last question, and then we can use whatever moments we have left for kind of an informal chat.
Yeah, sure. Thank you very much. That was a really good talk, Dr. Ben Maamar. I'm Alex. I'm a PhD student from Duke University, and I was just wondering, I may have missed this, but could you talk a little bit more about the background of the data that was used for the differential expression? Because I'm wondering, just for my own personal questions, like thinking about reproducibility, if this is a complex microbial community versus something like a host genome that's well annotated from a mouse, like you would probably see more consistency with these things.
Yes, and thank you very much actually for asking this question. It's very relevant. So the way we did this work so far was to focus on Escherichia coli genomes. And what we did basically was to get this, we took like about 3 most recent genome version of Escherichia coli, and we fragmented it like computationally in silico. So we created kind of a reads, something that you would have from a sequencer, for example. Except that in this case, since we started from a reference genome, we know exactly where each reads should go on the genome. We know exactly the position of all the genes and how things should be done. So by doing this approach, we had kind of a ground truth that we could use to compare the performance of other tools, like the aligners, for example. Since we know that this specific piece of sequence should be located here in the genome, we could see how the different aligners are performing, like submitting these reads that we created, and see how good it is at recognizing the right position of each reads. So this is one of the first thing that we did to both look for aligners and also counting steps. But the idea behind this work, obviously, we don't want to confine it to Escherichia coli. So one of the perspectives to this work is to expand it to Caenorhabditis abditis elegans, which is a eukaryote or worm that is very heavily used as a model organism in medical studies and biology in general. It's much more complex in that case because obviously eukaryotes are more complex. You have this alternative splicing of mRNA molecules that doesn't happen in prokaryotes. So it's going to be much more challenging to test, but this is like a perspective of the work. I hope I answered your question, Alexander, but let me know, please, if you want more details about specific method.
No, that was great. Thank you very much. Looking forward to the paper.
Thank you.
Okay, I'm gonna stop recording here.